It is my pleasure to present the Edwin G. Conklin Medal from the Society for 2017. First, I want to tell you a little bit about Conklin, and I'm not going to spend a lot of time on this, but he was an extraordinary scientist. His mentors, he was born in 1863 and died in 1952, and so he spanned a very critical period at sort of the beginnings of experimental developmental biology. And his mentors were committed to pure descriptive embryology, but he had this really sea change in which he incorporated experimental manipulation of embryos into his work. He was the director of Woods Hole for, I think, 35 years or something like that. And he worked on spiralian embryos, and he had the interesting idea of incubating them in different salt concentrations in seawater to look at what was going to happen to embryonic development. And he realized that he could use spiralian embryos to trace cells in the embryo and determine what they became. So his work was really foundational for the field of experimental developmental biology. But I want to spend a minute telling you about another aspect that I think was really important. So he had very heartfelt views about the role of scientists in society. And during his lifetime, he spanned the time of eugenics, and he was a champion of the ability of individuals to better themselves and contribute to society. And I just want you to see this quote from him in an address to the American Association for the Advancement of Science in 1937. He said that the ethics of science regards the search for truth as one of the highest duties of man. It regards noble human character as the finest product of evolution. It considers the service of all mankind as the universal good. It teaches that human nature and humane nurture may be improved, that reason may replace unreason, cooperation supplement competition, and the progress of the human race through future ages be promoted by intelligence and goodwill. I think this is a remarkable statement of, I think, what we should all strive for as biologists and scientists. And so we present this award in his honor, and this year our awardee is Philippe Soriano, who's at Mount Sinai. And preceding awards have gone to outstanding members of our society who have contributed in outstanding research in a sustained way over a long period of time. And Phil, of course, is no exception. But when I called him to tell him he had won this award, he was so surprised that I actually hung up the phone, and then I had to call him back to assure him that I wasn't kidding, because he actually did not believe that he had actually won the award. And I should stop here to say that I think Phil has scrubbed all pictures of himself from the internet. However, I have some secret sources that managed to find some interesting pictures of him. Once he got into his head that he had actually won this award, he decided, well, if the hat fits, I'll just wear it. So I have another picture of So I want to talk for a minute about what he has contributed to our field. So happily for us, he moved to the U.S. And came as a postdoctoral fellow to Rudolf Janish's lab, where he began thinking about retroviral insertions and the ability to use them or develop them as a tool in developmental biology. And I would say that his signature on the field has been to devise very sophisticated technical approaches that have then allowed him to make many mechanistic advances in understanding the mouse embryo, and in particularly in how signaling pathways work. So I just want to highlight a few of these. I think many of you are maybe aware of Phil's contributions, but his interest in retroviral insertions developed into these very sophisticated gene traps, and this was before the time when we could really, we had very few genes cloned and very little understanding of how genes might contribute to embryonic development. And Phil had this idea that he could use insertional mutagenesis to insert a reporter tag, and at that time it was beta-galactosidase, randomly into the genome, and then look at where those expression domains happened during embryogenesis. And from this screen, we recovered many genes that are really involved in early embryonic development, and it was our first ability to look at where they are and how they might be involved in morphogenesis from a single-cell embryo. And then he used also his, he was involved in some of the first targeted mutagenesis, and his interest in, I think, tyrosine kinases perhaps arose from targeting SRC very early on in one of the first targeted mutagenesis experiments that were ever done in the mouse. But then his gene traps just happened to entered randomly in the genome, but one of the genes that he trapped was the line called ROSA26. And many of you may recognize this name. It turned out, it came out of this random screen, but Phil recognized that this particular insertion trapped a gene that was ubiquitously expressed. And he realized that if he could make this conditional upon the promoter that was expressed to turn this trap on, then we would be able to use it for many, many applications in our experimental work. And this was developed into the ROSA26 Cree-based reporter lines that have spread to many other organisms, not just the mouse. And then I want to mention one other thing. So more recently, Phil has been working on downstream networks from signaling pathways, trying to understand how different receptor tyrosine kinase exert unique effects, even though they commonly use the same downstream signaling pathways. And so he's done very detailed mutagenesis of different parts of the tyrosine kinase receptor, disrupting different aspects of their signaling pathways. And he's used a novel screen to insert, to tag the signaling pathway networks downstream of these receptors so that he could look at how they behave when you disrupt the functions of the receptor. So it was really a very sophisticated screen. And what I want to emphasize is that it's not just that he builds these amazing technical tools for the field, but that he uses them to make important discoveries. And so here's a picture of Phil with his craniofacial friends here. So he's been very involved in understanding craniofacial development and how the receptor tyrosine kinases, like PDGF receptor and ephrins, have been involved in craniofacial development. And one of the people who wrote letters recommending him for this award commented that he has brought signaling out of the dish and into the embryo. And I think this is really true. He has designed the tools that allow us not to just look at signaling pathways in dishes, but to look at them in live embryos. So I also want to emphasize that another aspect of Phil's contributions is that he has this enormous enthusiasm for science that is both infectious and very inspiring to people who work with him and many others in the field. And he has been tireless in teaching and generously supplying his time and his reagents for anyone who needs them. And this is a picture of Phil with Janet Rassant and Robin Lovell-Badge teaching at a course in Mexico City. So he's been so important in influencing the next generation of developmental biologists. And I think that this is sort of in the spirit of Conklin, sort of contributing as a scientist should to the society around him. So maybe his good fortune is due to the oversight of the kitsune that he likes so much, and these little weird Japanese foxes that in Japanese mythology turn up very often, for good or bad. It turns out that they're a little bit unpredictable, and sometimes they do really bad things, and sometimes they do really good things. But I think Phil might attribute a lot of his success to the oversight of the kitsune around his house. But I think an introduction to Phil has to invite his chief protectors into the room. And this one, Arthur, took care of Phil almost the whole time he was in Seattle, and was, I'm sure, very important in contributing to Phil's work. And the new one, Waldo, has sort of taken over this job when Phil moved to New York, and is now doing his best to take care of Phil in New York. And what was really surprising to me is that just yesterday, I discovered that Conklin was born in Waldo, Ohio. And I think there must be something really weird about this. I don't know. But it's a great pleasure to introduce Phil Soriano and to award him the Conklin Prize this year.
Also, all the others who contributed to all the photographs. And actually, I know who some of the photos are. So I will talk with each one of you after the lecture. The second thing is that indeed it's an extraordinary honor to be receiving the Conklin Medal. I was indeed incredibly surprised, and I was overwhelmed, and I'm still overwhelmed. It's really a very distinguished award from what I'm going to have to say was a line used by Richard Harland when he received the Conklin Award, but which is from my favorite society. Richard, it can't be just your favorite society. It's also my favorite society, and I guess it's Maria's favorite society. So, and what I'd like to say is at the end, I will come back to why this is my favorite society. And then I was wondering, well, why do I receive the Conklin Medal, and what could I have had in common with Conklin? And then I started thinking about it a little bit more, and I realized, I hit the eureka moment, and I realized that Conklin and I did have something in common that actually Blanche did bring up, which was our fascination with marine invertebrates. And I might even have started a little bit earlier than Conklin. Conklin, of course, worked on cell lineages, on the slipper snail. I started when I was 2 years old. And when I was 2 years old, I found this shell on the beach. And I was really fascinated by the shell. I didn't want to lose it. And so I asked everybody, do you have a box? Because I don't want to lose the shell and so on. Nobody had a box. They wanted me to go. So I found a safe place for putting the shell. I put it into my ear. And that evening I had to be taken to the clinic to take it out. Okay. So I want to reassure you, I've actually grown out of this phase. I've stopped doing experiments on myself, I promise, okay? But I've maintained my interest in biology. I got turned on into genetics. I did my PhD in France, I'm half French, and I was working on genome structure. And it was when I did my postdoc, when I moved initially to Germany and then to Boston with Rudolf Janisch, that I got really turned on to developmental biology. So as I was saying, I was in both locations. This is Rudolf, a much younger Rudolf in 1987. And at that time, I started working with retroviruses as tags, as lineage tags. And we were working on a number of questions about founder cells, questions that were dear to the heart of my scientific grandmother, Beatrice Mintz, and that actually establishes yet one more point of lineage, which is a lineage relationship between Blanche and me, because Blanche trained with Beatrice as well. And then also working on insertional mutagenesis, and in this case, one of the genes that was disrupted was MOV34, which turned out to be a regulatory proteasome component, but Rudolf had previously disrupted collagen alpha-1 type 1 gene, and had found that basically this disruption was essentially the first knockout, and he did all these studies and so on. So I was really captivated by these ideas of insertional mutagenesis, and when I moved to Baylor for my first faculty appointment, I was there for 5 years, I continued pretty much on this theme. And so insertional mutagenesis, the idea at the time in the late 80s, was to really have a foreign piece of DNA that could integrate sort of anywhere, but it could of course integrate within another gene and cause a mutation in that gene, but you could use that foreign piece of DNA as a molecular tag for cloning the gene. There was no gene targeting really at that time. And so you could do it either by retroviruses. Retroviruses insert very cleanly. There's only a single copy. They insert usually at the 5' end of the gene, so they would be more able to cause mutation and null mutation in the gene. So they were easier to clone than microinjection transgenics, where the transgene would cause concatenamers and sometimes cause chromosomal rearrangements, but they could also be identified. Nonetheless, the efficiency was reasonably low, and we wanted to improve on this, and so our answer to this was essentially to build these gene traps, or promoter traps as we called them at that time. And the concept of these gene traps is shown over here, which is to introduce a reporter gene that essentially lacks a promoter, which is shown over here. We used, in this case, a beta-gal neofusion called beta-geo. And it would normally not be expressed, but if it integrates in the right frame and right orientation in a gene, it's going to be expressed. Because introns are bigger than exons, we put it downstream of a splice acceptor. And being a good Yanushite, or ex-Yanushite, we put it in a retroviral vector, but we put it in reverse orientation with a splice acceptor. And that's what ROSA stands for in all of these vectors. And so we made retroviral gene traps with this sort of a method. And when Rosa heard about this initially, she claimed that she was devastated because she thought she had a secret admirer. She had many. But she then realized what this really stood for. And these ROSA vectors typically cause null mutations in genes because the splice acceptor that we used in these vectors is very strong, has not been shown to be bypassed. And also, the retrovirus, as I mentioned, goes to the 5' end of the gene, so there's not a lot of protein upstream. And so with these types of vectors, what you can do is you can identify the flanking sequences by a PCR-type method. And so you can identify this gene that's mutated. You can go ahead and then look at gene expression. And let's say you want to do a screen based on only genes expressed on the limbs. You could just look at gene traps that are expressed on the limbs and analyze those. And last, of course, you could breed them to homozygosity and look at the phenotypes. And so when we did these sorts of studies, and this was all done at Baylor, we found these very, very cool expression patterns. These were the Spock mice. Okay, it turns out to be an insertion in the SEC8 gene, which we went on to characterize. And we had a number of other insertions which we obtained, which are shown over here in the original paper, some of which had restricted patterns of expression, but some of which, of course, are more ubiquitously expressed. And one of the ones that came out of this was ROSA 26, which I'll touch on later on. But as I mentioned, we wanted to breed these animals to homozygosity. So what we did is we did this, and I was expecting that all the lines would be embryonic lethal, but only 9 out of 24 lines were embryonic lethal. This was all done by Southern blots at the time. And this is because a lot of genes are not essential. We weren't accustomed to this idea. There's a lot of gene redundancy. If I had read the yeast literature at that time a little bit more carefully, I would have known that there would be a lot of gene redundancy, but this is what we found. And talking about gene redundancy, I wasn't even psychologically prepared to really deal with what I found afterwards, which was when we started using gene targeting to disrupt the SRC family kinases. So my interest in SRC actually goes back again to Whitehead, when I was a postdoc with Rudolph, because David Baltimore on the next floor had just identified this neuronal form of SRC. And I thought, well, SRC is important in all sorts of signaling pathways. It's going to be downstream of growth factor pathways and so on. We're certainly going to get a CNS-specific mutant and maybe a gastrulation phenotype or something like that. And instead, what we observed, and this is one of the first knockouts, as Blanche mentioned, we observed osteopetrosis. So what osteopetrosis is a bone phenotype, where essentially means stone bone, in which the bones become harder and denser because the osteoclasts fail to remodel the bone. So this was a bit surprising. We went on to show that the defect was actually cell autonomous to the osteoclast. And this shows over here on this slide, from the original paper, the failure of the bones to be remodeled in the SRC mutants. And it's a very serious mutation in the mouse for a rodent because they actually have so much bones that the incisors don't erupt. And so I had to feed them with my French background pâté, which is powdered food in baby formula with agar. But I can give anybody the recipe if they're interested. So we went on with this. Instead of just abandoning this, we went on to disrupt many other family members of the SRC family kinases. There's 8 family members. We disrupted 6 of them. And Fin and Yes are broadly expressed. And when we make compound mutants with Fin and Yes in the double and triple knockouts, we observed more severe phenotypes, and the triples became interesting because we could show that actually SRC family kinases are really essential for integrin signaling, but not in a way for growth factor signaling pathways. And then we also mutated Hick, Figure, and Lin with Harold Varmus and other members at UCSF to look at this in more detail. We then also disrupted a negative regulator of SRC family kinases, which is CSK. So all the SRC family kinases got activated, and those also led to embryonic lethality at day 9.5. And over the years, a number of people have worked on these projects. So at about this time, I had moved to the Hutch in Seattle, and I spent 15 years there. And those are the Fred Hutchinson buildings on Lake Union, and we had just moved to this location. And it's really at around that time that I started getting very interested in growth factor signaling pathways. And I got into growth factor signaling in a way through SRC. And again, looking at this very biochemically, and this is work from Sarah Courtenay, where she had found that not only SRC, but yes and FIN could also associate with the PDGF receptor. And this was one of the arguments for genetic redundancy, which was that if ever SRC wasn't there, FIN and yes could still do the role that SRC would have. And this was actually mentioned in the initial paper. Again, we weren't accustomed to thinking about genetic redundancy. So I was very interested in the PDGF receptor already on its own from this biochemical standpoint, but then I was also very interested in it from its background, from its phenotype. And the phenotype was sort of guessed at by this mouse, which is a classical mouse mutant called Patch, not Patched, Patch. And what you can see over here is the heterozygous phenotype in the Patch mutant, which is a pigmentation phenotype, which was already very interesting, but it turns out that this mutation is a 500 or 600 kb deletion that deletes the PDGF receptor alpha, but it also abuts the c-KIT gene. And so one of the questions was, is this phenotype only due to PDGF receptor alpha, or could it be due to some misexpression of the c-KIT gene? And on top of it, the null mutant phenotype of this patch mouse had been analyzed previously, well before, by Hans Grüneberg. Let's go back to 1960. And what Hans Grüneberg had found at that time is that when you breed these animals to homozygosity, you get what he calls these cleft-face monsters. This was my first encounter in craniofacial development. I was fascinated by this. And so I wanted to know, is this really PDGF receptor alpha or not? So what we decided to do was to actually disrupt this gene. What we found was most of the embryonic phenotypes associated with the patch mutant, but no pigmentation defects. That was shown subsequently by Peter Bessemer to be due to a misregulation of the c-KIT gene. And we could show subsequently also that the PDGF receptor alpha is working autonomously in the neural crest compartment. So we disrupted PDGF receptor alpha. We also worked on the PDGF receptor beta, and there we found a very different phenotype, which was more of a vascular phenotype. And so this already brought up one question, which was, why are there different phenotypes? Are these different phenotypes due to differences in the expression patterns, or are they due to intrinsic signaling differences? So what we decided to do were to do these knock-in experiments, where we introduced at the PDGF receptor alpha locus the beta kinase ability, and at the beta locus we introduced the alpha kinase activity. Because the receptors bind to ligands somewhat differently, we introduced chimeras, in this case at the alpha locus, which was the alpha extracellular domain hooked up to the beta kinase, and we did the converse experiment at the beta locus. And since these were all done by cDNAs, we also did wild-type cDNA controls, which developed perfectly fine. So in the homozygous state, these alpha-beta or beta-alpha mutants developed perfectly well, but in the hemizygous state, the beta-alpha mutants showed signs of vascular fragility. And this indicated that although the expression patterns were probably very important to explain the differences in the phenotypes, there probably were some intrinsic differences in the signaling abilities of these receptors. So we went on to analyze this in more detail, and the way that we started doing this was to do these allelic series of point mutations, initially at the alpha locus and then at the beta locus, where we disrupted, in the case of alpha, the ability of SRC to bind to the PDGF receptor alpha, the ability of PI3 kinase to bind, and the ability of all these other sites to bind as well. And so what we found from these types of studies, where we're specifically affecting one signaling pathway downstream of one receptor, is that PI3 kinase is the important pathway downstream of PDGF receptor alpha. But in the case of the PDGF receptor beta, where we built a larger allelic series of point mutations, we found that they were actually more additive type of pathway of all of these different signaling pathways contributing to the fate of pericytes that depend critically on PDGF receptor beta. And so what we found over here is that these different modes of signaling and a number of different people over the years have worked on PDGF receptor signaling pathways in the lab. I should point out that we had really a lot of alleles at this point. We had to genotype all of them. It got more and more complex. And when we reached 100,000 tails, my colleague Susan Parkhurst made this wonderful cake for us, showing the 100,000 tails. And what you may be able to see is at the base of the tail a very artistic and actually symbolic, and tasty I should say, use of the raspberry jam. And I'll let you think about what that actually means. So that was the 1-phosphate at a time part of the talk. But also we were trying to build a better mouse. And this is actually no joke. This original poster is in our tissue culture room, and it's been governing all of our point mutations over the past 30 years. So coming back to Conklin, Conklin also worked on sea squirts, and he had found that actually in the Stiella, a single blastomere was pigmented, and the descendants actually were also pigmented. So in a way, he was using one of the first cell lineage markers, and our answer to this was really to build the Rosa 26 Cree reporter mice, which Blanche touched upon, which are essentially shown in this diagram below. And so what you can see here is that basically the idea is that you're using a ubiquitous promoter driving LAC-Z, but its expression is prevented by a lock-stop-lock. So even if Cree is active in a single cell, all of the descendants of that cell will still express LAC-Z because the promoter is ubiquitous. So this is a lineage trace method, and it's also can be used for testing Cree lines. It's also a very good locus for expressing all sorts of genes. And one of the great things about Rosa 26, and here I'm going to retaliate in terms of the photographs, is that I actually have a publicist. And the publicist is actually Richard Berenger, who likes to tweet about this. Richard, there's actually 870 lines now, of which 680 have been targeted alleles. So we were actually interested in using the gene traps for really trying to look at PDGF target genes, and the problem was that ES cells were not very responsive to PDGF signaling, as Kat knows well. And so what we decided to do was to combine gene traps with microarrays. And as shown in this sort of a diagram, this was done by Weisheng Shen in my lab. And using this sort of a screen, what essentially we did with a gene trap library and building arrays of mutant mice in a way, we were able to identify a number of different PDGF target genes and to build a lot of different gene traps over the years. So I mentioned Rosa 26. I should mention Rosa 53, which is the shroom mutation in which an actin-binding protein is disrupted, and this affects the polarity within the central nervous system. And also with the gene trap array, we found a number of different phenotypes that were not only confirmatory, but also predictive of PDGF receptor activity when we went back to the PDGF mutants. And so over the years, a number of people have worked on gene trap lines, and I just want to mention also that we've worked over all these years on a number of other projects that I don't really have time to touch on. But when Frank Gertler was in the lab, we worked on MINA and cytoskeletal regulation, on ventral folding morphogenesis with Masayuki Komada, on SMAD signaling with José Aubin, and on ephrin signaling, both forward and reverse signaling, when Alice Davy and when Jeff Bush were in the laboratory. So at around this time, we had moved to New York. There's another tower that has actually gone up next to this big dark tower where I work, but it's over Central Park, so that's quite nice. And it's at this time that we started working on FGF signaling. And what I'd like to do in the remaining time during this talk is give you a little bit of a vignette of the more recent work that we've been doing in the laboratory and where we're really thinking and what we're trying to do. And so I'd like to talk now about FGF signaling, and there's many different FGF ligands, and there's 4 different FGF receptors, of which really 2 play very critical roles in early embryonic development, which are FGFR1 and FGFR2. FGFR1 was long known to play a critical role in EMT at gastrulation. This is work that Brian Ceruna did when he was with Janet Rossent. But it was also known that FGF signaling played a very important role also in early embryonic development, which is in the pre-implantation embryo. Kat talked about this yesterday, Kat Jantanakis, in her talk yesterday morning, and essentially it's in the second cell fate decision in the embryo, which is to form these blue cells over here, which are the primitive endoderm, which give rise to components of the yolk sac, and also to segregate these from the pluripotent epiblast over here in red, which forms the early embryo. So it was known that FGF4, the ligand, plays a role in this process, but it was initially thought it would be FGFR2 that would be playing a role based on its expression pattern. This, in a way, didn't really make much sense because FGFR2 is lethal at much later stages of development, even if there were some debates about the FGFR2 null mutant phenotype. This has been put to rest now. And we had found that putting the FGFR1 null, analyzing this on a 129 co-isogenic background, led to lethality at E5.5, and what we found here is a reduction in the number of primitive endoderm cells. And so what both Kat's lab and our lab have shown is that it's a combined action of FGFR1 and FGFR2, which is critical for the formation of the primitive endoderm, with FGFR1 playing an important role in this process, and Kat talked about this yesterday. So over the years, a number of people have worked on different aspects of FGF signaling. And I wanted to discuss this primitive endoderm work because I want to discuss the parallels with what happens in craniofacial development. So in craniofacial development, if you disrupt FGFR1 in the neural crest compartment, what you can see over here with the WIT1-Cree driver, you get this very significant facial cleft. But the phenotypes that were found with the FGF ligands, again, FGF8, turned out to be more important. And so we thought, well, maybe there's another receptor again, which is playing a role. And so we thought, well, maybe it's FGFR2. And indeed, if we start looking at the combined actions of both FGFR1 and FGFR2, what you can see over here is that FGFR2 alone doesn't lead to a facial cleft. And even if you remove one copy of FGFR1, it's not more severe. This is what I showed you before for FGFR1 alone. You remove one copy of FGFR2, this is actually much more severe than what you see here, and you remove the two receptors altogether, and you get this very, very dramatic phenotype with agenesis of all the frontal bones. And likewise, there's a very severe phenotype in terms of mandible development. So again, what's happening in this case is that it's both FGFR1 and FGFR2 which are playing a critical role in this process, with, again, FGFR1 playing the most important role. So we went on to try to look at what's going on with this. We find that there's a general rate of proliferation which is decreased in these FGFR1, FGFR2 mutants. And what we find is that we find very increased apoptosis in a part of the frontonasal complex, which are on the lateral side of the nasal pit, which is here, which is called the lateral nasal process. And so to simplify a little bit what's going on, what we can think about over here is the in terms of craniofacial morphogenesis, is that you have the maxillary process and the mandibular process, which are shown over here in yellow and in orange, and this frontonasal process, when the nasal pits are formed, are separated into this medionasal process. That's actually where we find a defect in cell proliferation for both the PDGF receptor alpha and a downstream target, SRF, that we've analyzed in the laboratory. But what we find in the case of FGFR1-2 is increased cell death in the lateral nasal process, which is shown over here in purple. And it's known that this lateral nasal process and the maxillary are structures that actually come together and bring all these primordia together during embryonic development. So this might be a different mechanism for how these facial clefts are coming about. So we also like to study how this works in terms of cell signaling. And you knew I wasn't going to drop signaling so quickly. And so let me tell you a little bit about cell signaling. So the main mechanism by which FGFs are thought to signal is to signal through ERK signaling over here. And this is shown in this slide, where essentially the FGF receptor is going to go through the FRS2, GRAB2, RAS, RAF, MEK, ERK signaling pathway. It's also known that ERK signaling can be engaged by a number of other pathways. But FGF isn't only an ERK-dependent pathway. FGFs can also engage a number of other signaling pathways, such as PI3 kinase, through FRS2, and also probably through some other pathways, and all these other pathways as well in mechanisms that are less well understood. And so to try to understand a little bit how this works, we developed over the years in the laboratory, again, a allelic series of point mutations for both FGFR1 and for FGFR2, that disrupt the ability of CRAC to bind to the FGF receptors. We call these mice C. To disrupt the ability of CRAC, phospholipase C-gamma, and GRAB14 from binding to the receptors, CPG. To disrupt the ability of FRS2 from binding to the receptor, call these mice F. All of these mutations together, so we call these mice FCPG, and we compare them to the null. And so I'll just show you the data for FGFR1. FGFR1, again, on this 129 background, die at E5.5 of development. They have a primitive endoderm defect. The C and the CPG mutants are viable. The F mutants are lethal at day 0, and they have these skeletal defects and cleft palate defects. So there's a number of processes which are probably working in an FRS2 independent fashion. And when we make all the mutations together, we get this phenotype at day 10.5 of development, which is embryonic lethal, very severe, posterior truncation, but they actually gastrulate, they form somites. And so we're not recapitulating the null mutant phenotype. And so if we start looking at the downstream signaling pathways, just looking at ERK over here, what you can see is that ERK is severely affected when we disrupt FRS2, almost totally abrogated when we disrupt FCPG, but other signaling pathways are not affected, particularly PI3 kinase. And so we think that we're missing some effectors. And this is the story for FGFR1, and this is the story for FGFR2. And so we're using proteomic approaches to try to look at this in more detail. But coming back to craniofacial development, if we then make combinations of mutants for both FGFR1 and FGFR2 carrying the mutation in the FRS2 binding, at least in this combination of phenotypes over here, where it's homozygous for the FGFR1 mutation and heterozygous for the FGFR2 mutation, you can see over here this facial cleft at day 14 of development. And this, therefore, says that ERK is important for this pathway. So what I've told you so far is that we have two receptors that regulate craniofacial development, with FGFR1 signaling through the ERK signaling pathway and PDGF receptor alpha going more through the PI3 kinase signaling pathway. And so this really brings up the last point, scientific point that I want to bring up, which is what are the parameters that really encode transcriptional specificity downstream of these RTKs? And so you could think that it's maybe a question of qualitatively different effector pathway, with PDGF receptor alpha being a more PI3 kinase-ish type receptor, and FGFR1 being a more ERK-ish type of receptor, and maybe some other signaling pathways being involved. But when Harish Vasudevan was in the lab, I told him this old story of PC12 cells. And so we're going back 22 years with this. And the story here was that when you take PC12 cells, and if you treat them with EGF, you get this rapid pulse of ERK signaling, and it makes these cells proliferate. But if you treat them with NGF, what you get is you get a long duration of ERK signaling, and it makes these cells differentiate. And this there was sort of summarized in this very beautiful article by Chris Marshall, that it was really signal duration or signal magnitude that could be just as important as just the nature of what you're really trying to, or the signaling pathway that you're engaging. So Harish wanted to know, does this happen also in craniofacial development? He isolated from palatal shelves primary mouse embryonal palatal mesenchyme cells. What he found is that when he treats them with FGF or with PDGF, you get a long duration of ERK signaling in the case of FGF, a short duration of ERK signaling in the case of PDGF. There's no big difference in terms of AKT signaling downstream of either one of these receptors. He did RNA-seq, and what he found is that FGF regulates a number of genes involved in cell proliferation. PDGF regulates genes which are more involved in cell differentiation. And going on with this, he wanted to go ahead and test it in these cells, in the MEPM cells. And indeed, FGF is more proliferative than PDGF. And if you go and look at cell differentiation, what you can see is that when you treat the cells with PDGF, you get these alkaline phosphatase cells that emerge, whereas you don't see this with FGF. If you then go ahead and do the assay, but also inhibit on top of it the MEK signaling pathway, so ERK, or you inhibit the PI3 kinase signaling pathway, you can see that if you inhibit the MEK signaling pathway, you find all of these differentiation which occurs, and you don't see that in the case of PI3 kinase. So to summarize what I've told you over here is that we think that we have two different pathways in these MEPM cells. It's probably very context dependent, where FGF is actually promoting cell proliferation, blocking cell differentiation, and PDGF is favorizing cell differentiation. And if you inhibit the ERK signaling pathway, what you find is that you get cell differentiation. If you block PI3 kinase, you block cell differentiation. So this was really what I wanted to tell you, which is really a very long saga of how we were trying to build a better mouse, one phosphate at a time, and where we're ending up now is with this story of trying to really look at ERK signaling itself and PI3 kinase signaling, and we're looking at that, we're looking at it by signaling dynamics, and we're looking at it also with biosensors. And so with this long saga, which I've told you about, it's really only been possible to accomplish because I've worked with really, really wonderful individuals over the years. It's really been a privilege to have such great people. They actually make it all possible. And also it's wonderful to see them develop and to become successful investigators on their own. This is already a theme which we've heard in the previous talks. And so what I think is also very comforting for me is to learn that this award was actually given to me following a nomination, I think, from a number of my former trainees. So I'm incredibly touched by that. It's incredibly heartwarming. And also by a number of scientists who I've had the honor of working with over the years. So that is really the really, really special thing. I think when you see people who do well and they return it, it's just absolutely fantastic. And I promised that I would tell you why SDB is my favorite society. So I just want to close on that. And really it's my favorite society because they really care about the same, they have those values. They really care about that. They really care about training. They really care about mentoring. You had this amazing postdoctoral symposium a couple of days ago. This is what all the society is about, and really what everything is for developmental biology is the future, and all the young people who are over here. So I'd like to just close over here and thank you all for this absolutely extraordinary award. Very honored.